Welcome to the Atlantico podcast, where we talk about the science behind the Atlantico project, the Atlantic Ocean, and the human adventures experienced along the way. Here, we have conversations with guests from around the world who work together so that we can better understand, manage, and protect the ocean. So let's embark on the journey of Atlantico and discover the world that lies above and beneath the surface of the beautiful Atlantic Ocean. In today's episode of the Atlantico podcast, we have the pleasure to have a conversation about plastics in the ocean and the life which develops on it, the so-called Plastisphere, with our guest, Linda Amaral-Zettler. Linda, hi and welcome to the podcast.
Hi, thank you, Eloise.
Before we talk about plastics, could you maybe introduce yourself and tell our listeners about what you do and what brought you into your line of research?
My name is Linda Amaral-Zettler, and I'm a research leader here at NIOS, which is the Royal Netherlands Institute for Sea Research. And we're located on the island of Texel in North Holland, in the Netherlands. I am also a professor at the University of Amsterdam, where I hold a chair in marine microbiology. And this gives me an opportunity to teach. I co-teach a course in microbial ecology. And I'm also able to advise PhDs and master's students. So I pretty much spent my entire career as a researcher in the United States in the village of Woods Hole, Massachusetts, which is home to many scientific institutions, until I moved my research group about 5 years ago to NIOS. And I still have an affiliation with my position at the Marine Biological Laboratory. A major focus of my research is really looking at biodiversity of microbial life, and in particular, using approaches that are called molecular approaches to study the diversity of microbial life in the ocean. And a lot of microbes can't be differentiated based on their looks, so to speak, their morphology. So we use these molecular approaches, sequencing of DNA, to describe the relationships between these single-celled organisms that are otherwise challenging to differentiate. And what's often called the gold standard for genes to target for this kind of approach is a gene that codes for ribosomal RNA.
Can you tell us a bit more about that, what it means?
Yeah, definitely. Absolutely. Yeah. So one of the fundamental building blocks of ribosomes is ribosomal RNA. And the ribosomes are the protein building machinery of cells in our body and in all life, really. And I really started my career in marine microbiology when these molecular techniques were to describe the diversity of microbes in the ocean were really just starting to be applied whole scale to study diversity of microbes in the ocean. And so a turning point in my career was what is called the first census of marine microbes. And I was the program manager for this program. I also served as the education and outreach lead. And it was a program that was funded through the Sloan Foundation. It was global in extent. And it was co-directed actually by scientific leaders, Mitch Sogan, who is at the Marine Biological Laboratory, and Jan de Leeuw, who is ironically at NIOS now. So both my former and my current research homes. And the focus of this first census of marine life, which was called the International Census of Marine Microbes, was largely on microbes that live in the water, particularly the water, what we call the water column, so the floating part of the ocean, but also in the sediments. And towards the end of this period is when my group started working on microbes that weren't so-called free living, but attached to surfaces in the ocean. And we focused on what we call a biotic surface, which was the floating macroalga called sargassum, which is the namesake of the Sargasso Sea that surrounds Bermuda, and also a new substrate in the ocean, and that was plastic marine debris. So that's sort of how I came to start studying plastic, and in particular, life that lives on plastic in the ocean.
Brilliant. That sounds really interesting. Can't wait to hear more. Before we dive into that sort of subject, can you tell us, how did you get involved in Atlantico?
Sure. I like the story because I think it's quite fitting and almost ironic in that I was on a 5-week cruise in the South Atlantic sampling what is now called the Great Atlantic Sargassum Belt. And I mentioned sargassum a minute ago. This is a brown macroalga. And unlike many of the species in this genus, it spends its entire life cycle floating in the ocean, particularly in these open areas of the ocean. But recently, after 2011, it started increasing in biomass. And this region, this belt, is located between 5 degrees north and 5 degrees south of the equator. And it's normally a very unique and interesting ecosystem and a safe haven, honestly, for many protected species, and even has its own endemic fauna. But now it's stranding on 3 different continents and really wreaking economic and ecological havoc. So I was on this cruise and I was contacted by Daniele, and he was very keen to include not only plastics in the Atlantico program, but also the life that lives on plastic, what we call the microbiome of plastic marine debris. And sadly enough, this ecosystem that I'm referring to, the sargassum ecosystem, is very well endowed also with plastic debris. So you find, flip-flops and all kinds of plastic out in the middle of nowhere in these huge sort of what we call rafts of algae that are ecosystems in their own right, also, unfortunately, living the legacy of plastic debris.
That is unfortunate, and I guess we see more and more of that in the ocean. One of our main research focus in Atlantico is, as you said, microbiomes, but also plastics and the Plastisphere. Maybe you can tell us more about, firstly, maybe the issue of plastics in the ocean.
Well, I think nowadays there are very few people who haven't heard about plastic pollution in the ocean. It's really all over the media. And, I'm a microbial ecologist and have been studying microbes for years. And very few people get excited about microbes. However, when you start talking about microbes on plastic, suddenly people can relate. Because let's face it, plastic is pervasive in our lives, and it has really greatly improved our quality of life and certainly our longevity. However, plastic these days is mostly getting bad press. And the reason for that, of course, is because the increase in plastic pollution and the increase in plastic pollution awareness, particularly in our oceans. But some of this is sort of tainted, if you will, with myth regarding the extent and the intensity of plastic pollution. And so I think the problem with the concept is that there's this notion that we can merely clean up the garbage that's out there as if we were picking up, garbage curbside. And sadly, it unfortunately doesn't work that way. And that's not to say that beach cleanups aren't effective. They do, in fact, make a difference. In fact, the reason for that partly is because a lot of the plastic debris that enters into the ocean comes from land, not surprisingly. Some of it does come from fishing activities and, catastrophic events like tsunamis and hurricanes. But for the most part, it comes from land. And in 2010 alone, it was estimated that between 5 and almost 13 metric tons of plastic enter the oceans. Again, with the largest amounts, really the result of poor waste infrastructure. So this sort of concept of these floating islands isn't really accurate. Instead, we speak of, instead of speaking of garbage patches, the sort of plastic problem is often referred to as the plastic soup, because in terms of numbers, the amount of plastic that's in the ocean is mainly less than 5 millimeters in size. And it's sometimes referred to as microplastic or large microplastics, depending on the sort of latest definitions. So getting back now, transitioning a little bit away from the plastic pollution problem to studying what lives on this plastic pollution, we actually first got our very first samples that we characterized both using molecular approaches that I've just described, but then also microscopy, so very high-powered microscopy. These very first samples came from a cruise that, in fact, was trying to define the easternmost edge of the garbage patch, not in the Pacific, which I think is perhaps the most famous one and the largest one, but in the Atlantic. And this is where our very first samples that we characterized came from. And just sort of as a sad, what I consider a sad anecdote, this cruise went as far east as it could go, and we're still seeing some of the highest concentrations of plastic that they were able to count, or not even able to count effectively in their nets.
There's no edge, basically. Is that what you're saying?
There's no edge. There's definitely a concentration, but given the constraints of this cruise, they had to turn around before they could really stop seeing plastic in the ocean. And and so this was the first glimpse of what we call the Plastisphere, was this sort of dedicated cruise to find the eastern edge of the great Atlantic, or maybe not so great Atlantic garbage patch.
Can you tell us, why is it important to study the Plastisphere? What does it tell us?
Yeah, so let me first, I guess, just give you a little bit of background on what we call the Plastisphere. And really the analogy here is that it's the thin layer of life on the outside of plastic, analogous to the thin layer of life on planet Earth. So the biosphere on planet Earth is really a very thin layer that is alive. And so we likened it to that. And in fact, we now know that the Plastisphere is pervasive. There's really no plastic in the ocean that doesn't become part of the Plastisphere, so to speak, or that doesn't possess a Plastisphere. Microbes almost immediately adhere to plastic surfaces and form this biofilm, which is composed of many different forms of life, bacteria and archaea to a lesser extent, but also organisms that are related to ourselves, eukaryotes. So we like to sort of study this environment, this ecosystem, if you will, albeit new. We like to study it as a whole. So we study all the members. Now, the Plastisphere is not unique to the ocean, just like sadly plastic litter is not unique to the ocean. However, where plastic and the Plastisphere begins to become important is in the fact that most plastic litter is composed of plastic that floats. So these are single-use, often single-use plastics like polyethylene or polypropylene that are less dense than seawater. So they float. And because they are capable of floating, they can move with the ocean currents and get transported very large distances. So in some of our earliest studies of the Plastisphere, we made some really important observations that the Plastisphere could actually contain some unsavory microbes, in particular ones that can potentially cause disease in humans and other animals. And even prior to our own work, there were groups that were reporting things like harmful algae that can produce toxins that were becoming associated with plastic litter. So they can hitch a ride on plastic and go to places where they're normally not found. And, we've known about the ability of things like driftwood and plastic itself to transport organisms very long distances. But the sort of communities that have lived on these entities haven't really been very well characterized. And you can imagine that there are implications for aquaculture, for fisheries, and even for coral reefs, because, again, the plastic itself, in addition to, in some cases, depending on how large it is, kind of suffocating some of these types of environments. I'm thinking now of coral reefs. Again, if they contain organisms that are potential pathogens, these pathogens can be transported and subsequently cause disease in areas where that disease didn't exist prior. On the other hand, other investigators are really interested in studying the plastisphere because they are driven to find, novelty, unique enzymes, perhaps, that allow microbes to live on plastic and maybe even live off of plastic. And this motivation is often couched in the concept of microbes solving our plastic pollution problem. But I just want to say that I do not in any way, shape, or form endorse this concept. I think there is as plastic itself, a pervasive problem or misconception that eventually microbes will degrade our plastic waste. And what we need to keep in mind is that the timescales that this occurs is, they're lifetimes, they're hundreds of years, even more than lifetimes. And so we're leaving behind this legacy that I think no one wants to own up to, so to speak. And, we are looking for alternatives that are friendlier, environmentally friendlier. And there are candidates out there to conventional fossil fuel-based plastics. And there's certainly room for this in our economy as we wean ourselves perhaps off of the single-use plastic that is based on fossil fuels to reach for greener alternatives. But I think an important take-home message is that we always need to remember that the ocean isn't a trash can, and it's not even a composter, and that littering is never good anywhere, anytime. So the plastisphere is, I think, important to study for sort of its virtues and its vices, so to speak.
Extremely important. Thank you for that insight. Moving into maybe a more focused question about AtlantECO and to understand maybe how we are studying plastics and the plastisphere in AtlantECO. What are we looking for and where are we looking for these things?
Well, just sort of circling back maybe to sort of where the conversation began, we are really using methods that were developed to study the census of marine life, specifically the census of marine microbes. And these are these short fragments of ribosomal RNA genes that allow us to capture the diversity that lives in the plastisphere. So we're using these methods. And when we collect our samples, we don't only ask sort of who's there by using these molecular approaches, but we also try to identify the plastic, learn about the chemistry of the plastic, how weathered it is, how sort of affected by wave action or UV it might be. And then if there's enough sort of sample remaining, we also try to visualize the communities that live on the plastic. And sort of traditional ways of doing that uses something called scanning electron microscopy. And this is a very high-powered way of examining the communities. And the reason we like to sort of use very high-powered microscopy to examine the communities is because it tells us something not only about who's there, but their orientations, how they're associating with each other. There are now ways of combining some of the molecular approaches that we use with high-powered microscopy to start to understand not only the sort of morphology and the spatial orientation of these microbes, but also which microbes are associating with each other in this plastisphere community. And so that's a sort of an exciting new horizon, if you will, in terms of the kinds of ways we can understand what the membership of the plastisphere is like, and also how they're potentially associating with each other. In addition to sort of understanding who is present in these communities, which species or genotypes are present, we're also not only looking at what we call these marker genes that are telling us about the diversity present, but we're also looking at all the rest of the DNA that tells us about the metabolism of the organisms and their potentially ability to survive in this somewhat harsh environment. Because, out there in the Atlantic, in the open ocean, there's a lot of UV presence, there's wave action, it's a relatively harsh environment. And so better understanding how microbes cope in this environment, I think, is, again, interesting from a scientific perspective, but it may also lead to some interesting discoveries with respect to the enzymes that they possess and how ultimately we might benefit from them in that respect. And in terms of what we sort of have for samples to study within the context of Atlantico, my group has been really studying the problem since about 2010, so over 10 years now, over a decade. And so as a part of that sort of ongoing research, we've amassed many different samples from many cruises that we've conducted, both in the North and the South Atlantic and parts of the Caribbean, et cetera. So now we are sort of using the methods that I just described to really characterize these samples to the fullest extent possible, and then trying to use some of the approaches that the different work packages within Atlantico are developing to, better understand the implications of interactions between the microbes that are present in these communities, and ultimately potentially how these communities are in turn affecting the microbes that are living in the water column or in the sediments. And so that's sort of a summary of how we're focusing on the Plastisphere, at least in my group, in the Atlantico project.
Yeah, and in some of the expeditions that we have ongoing at the moment, so collecting new samples, these expeditions are going towards the river plumes and maybe up some rivers to try and maybe identify the origin of those plastics that we find in the open ocean.
Right. Yeah, that's sort of an unknown question, is what the overlap might be between organisms that are primarily fresh water that are coming from these land-based sources like rivers, of course, and to what extent they are then transported further into estuarine, brackish waters, and then ultimately full strength seawater. I think it's a really interesting question. Many microbes, in addition to being able to kind of be active at any point in time, can also form cysts. They can form spores. And so it might be the case that they might not be active immediately, but they can become active if the conditions change and they are allowed to live or be present in an environment where they can survive and thrive. So there's some really interesting questions that remain, certainly many interesting questions that remain with respect to plastics in the Plastisphere. And I'm excited about being part of Atlantico because I think it will really give us a chance to answer some of these unknowns.
That's brilliant. And I am sure that in future episodes of the podcast, we will come back with many more questions and hopefully some beginning of answers, or even answers about the microbes, the plastics in the open oceans, where they come from, what do they do, how do they evolve, how do they survive, all of that. Really exciting topic to cover in the future as well. Linda, thank you so much for the conversation today and for your time. I can't wait to hear some more in later episodes.
Thank you, Eloise.
We hope that you've enjoyed today's episode and look forward to seeing you next time. You can follow the Atlantico project on our website on www.atlantico.eu, and you can also find us on Twitter, Instagram and Facebook. All the links and information on the project and on today's episode is in the show notes.